First off, I would like to thank all of you for your interest in using our technology to improve lake water quality. My name is Gary Anderson, and I am the Chief Technical Officer of Veracet, a DNA testing company that provides smarter lake management through innovative methods in microbiome technology. I also have a joint appointment as a professor of environmental science at UC Berkeley and a senior scientist at Lawrence Berkeley National Laboratory. Today I will be talking about a scalable DNA-based approach to identifying all microbial contamination in watersheds. Many of you have noticed a recent increase in inland lake closures due to microbial contamination. These sources include fecal input of unknown origin. Also, nutrient and climate factors combine to create conditions that are favorable to contamination from harmful algal blooms. In these cases, there are unhealthy as well as undesirable conditions that limit recreational opportunities and overall enjoyment for the homeowner, as well as threaten their considerable investment. Typical examples would include fecal contamination from failing septic systems or changes in nearby ag management. You need to identify the culprit with absolute certainty for any hope of fixing the problem and restoring lake water quality. The problem is that existing tests are not able to identify with sufficient confidence where the specific source of pollution is coming from. Even more importantly, they're known to be very inaccurate, with many false positives and false negatives. This leads to a very large economic losses due to lack of clarity where and when the actual problem is happening. Late summer harmful algal blooms produce toxins that are very harmful to humans and their pets. No one wants to look out at their lake on a nice summer day and know that they can't even touch the water. HABs in freshwater lakes are primarily caused by cyanobacteria, also known as blue-green algae, that grow in response to various nutrients and environmental conditions. So how do we reduce the possibility of developing these algal blooms? By the triangle of monitoring, mitigation, and treatment. Current monitoring methods have not been particularly successful in reducing HAB. This is where we need to focus our efforts. So I would like to introduce you to the concept of total lake health. Recent advances in DNA technology give us the power to do what was considered impossible even a few years ago. We now have the ability to monitor the entire microbial community, the lake water microbiome, at an affordable price. Using this technology to monitor the water microbiome allows us to check the overall water quality. In other words, is the lake healthy? Identify any fecal contamination, or is the lake getting sick? Develop an early warning system to alert for potential harmful algal blooms. Measure the effectiveness of a treatment. In other words, is the lake returning to a healthy state? We have all heard about the human microbiome and how it relates to our personal health. This is something that we can apply to our lakes as well. It's a new concept for most, but gives us a new weapon to more effectively manage our lakes. Just so that we're all on the same page, let's take a couple of minutes to define what we mean by the lake water microbiome. I will attempt to illustrate this in the following simplistic representation of a cross-section of a typical lake. I have included a number of different styles of green shapes to represent the diversity of microbes that are always present in a healthy lake, as shown here. In reality, there could be over 10,000 different species of microbes that are present, each of these in a very, very small quantity in the lake water. These are not harmful, these are not pathogens, they are simply indicative of pristine water. In addition to these healthy lake microbes, a number of different algal species may be present, as indicated by these blue symbols here. Some may grow better than others under certain conditions or certain weather patterns, and a subset of these can produce various toxins. In addition, some microbes may grow under specific environmental conditions and are only found when certain chemical or physical factors are present. These are shown here as purple symbols. If there is contamination from a specific source, say horse fecal pollution, then we are able to identify hundreds to over 1,000 specific microbes that are particular to that source, as represented here with these horse figures. Similarly, if a human source, such as septic waste, is present, there will be hundreds to thousands of microbes specific to this source, as we see here with these red symbols. Finally, if a fecal source is present, then a very small fraction of these will contain pathogens that could be harmful to humans. as indicated by these symbols. Although it is important to remember that this is an extremely small fraction of the overall concentration of total microbes in the water. This very large and diverse collection of microbes in the lake water constitutes its water microbiome. And that is our focus at Veracet. We can detect all sources of contamination in one test using microbiome DNA. Our phylochip DNA microarray has the unique ability to rapidly identify all microbes in a single small sample of water. We are ultimately a DNA testing company, and we are able to use our technology to identify tens of thousands of different types of bacteria in every water sample. The power of the microbiome to identify the health or specific type of contamination that is present in a particular water sample is through a database listing all types of microbes that are specific to an individual contamination source or that serve as a sentinel of runaway growth of toxic algae. We are similar to DNA testing companies like Ancestry.com in that as we grow our database of samples, we increase the accuracy and resolution of our results. We have already spent a considerable amount of time collecting samples from different animal sources. We have also analyzed samples from different types of human sources, including sewage, septic waste, and fresh human waste as examples. Our methods can actually distinguish between these different types of human pollution. As you see, we've also collected and analyzed sources from a variety of domestic and farm animals, wild animals, birds, etc. We are now up to around 40 different types of sources that we can use as a baseline to detect their presence. We have been successful in using this technology to solve a number of real-world problems. For example, in Malibu, we were able to identify the source of contamination affecting the local beaches. We were able to identify that wild animals and not humans were responsible for watershed contamination in Kauai. In all these cases, we were able to bring clarity when people had been arguing with each other for a long time about who was responsible. The Russian River is considered an impaired recreational river of Northern California by the EPA. Here we see a map where the main blue line is the Russian River that flows into the Pacific north of San Francisco. What is circled in red is the location where we found the primary pollution on the river caused by failed septic systems from overused vacation homes. The green circle shows the location of dairies where cattle signatures were found on this tributary. The purple arrow points to where the river meets the sea, and huge number of seagulls here were the reason we detected this signature. What is really interesting is that all along the upper half of the river, the black dots were locations where we were able to show that this area was free from all contamination and would be considered healthy, even though it had some of the highest fecal coliform counts. What happens when you have a crowd of people in or next to a river for 2 days to listen to a jazz festival? We found out that you may not want to know the answer to that. As you see from this graph, a morning water sample was taken for 5 days in a row, including before and after the 2-day concert. The red bar on Sunday indicates significant human fecal contamination after the first day of the concert. The even higher red bar on Monday represents one of the highest levels of contamination we have ever measured. The standard fecal indicator test did not show any actionable levels of bacteria until Monday, when it was just over the threshold. When we compare our method of using hundreds to over 1,000 independent measurements to identify a source of pollution with statistical precision compared with the conventional methods that rely on one single microbe that potentially indicates contamination, we find that the current methods are wrong an alarming percentage of the time. In the graph shown here, we compared the values from a standard E. coli fecal indicator test, shown here on the x-axis, with a measured level of E. coli that was obtained from the phylochip on the y-axis for each of about 60 water samples. The samples where we found contamination by human waste are indicated by red squares, or by cows are indicated by triangles. The samples where phylochip identified a healthy water microbiome are shown as black circles. Overall, as the E. coli signal increases by the standard tests, it also increases by the phylochip, showing agreement of the E. coli values in both types of test. Anything to the right of the dashed blue line represents a water sample where the conventional E. coli test would be positive for the contaminated water. To the left would be a negative by the conventional test. By the colors on both sides of the line, we see that the actual results from the phylochip show contamination both above and below this threshold, as well as a lot of black circles, healthies, above the threshold. This is why we do not trust the results from the single fecal indicator tests. Although most of our work to date has focused on large watershed scale projects, Veracet sees the value of using its technology to aid in lake water quality management. The challenge is having a user that has no formal water testing training taking a single lake water sample for analysis. The methods required to collect the sample are actually reasonably straightforward, and shown here the first step is to collect the 100 milliliter water sample and then to concentrate the microbes onto a filter. Again, showing the first steps are to collect the water sample and then to concentrate by passing it through a filter. The reason for the filter is that it is easier to mail a small filter than a larger water sample. The next step is to send the sample by overnight mail to Veracet, or if you happen to be doing several samples at different times, you can place them in any freezer until you are ready to ship. Veracet provides a kit containing a collection syringe and filter to individuals that want to take a sample. So what do you get back when you send in a water sample? Here I am showing a mock sample report that has all of the pertinent information about the water sample and whether or not it passed quality control. In this example, the water sample had contamination from septic waste, as well as a lower concentration potential signal from geese. We are still figuring out how to display this for someone that wants to analyze just a single water sample, but one thing we are thinking about is color coding, so red for a statistically significant level of contamination, yellow for potential contamination, something that may pose a problem in the future, but does not rise to statistical confirmation at this point, and green, meaning that you have a healthy water microbiome. We could also include bar charts to signify the relative amount of contamination. Similar to genealogy-based DNA testing companies like 23andMe, we actually possess a huge amount of information that could be mined from each sample. For instance, you could figure out what types of algal species are present or how the water microbiome changed after a chemical treatment. Freshwater harmful algal blooms are often caused by cyanobacteria, also known as blue-green algae. Some, but not all species of these algae produce toxins, such as microcystin, that are harmful to humans and animals, but it is not clear what triggers them to suddenly produce the toxin. Our phylochip can detect 150 different species of blue-green algae, and it may be possible to use this information to help inform when to treat. The figure here shows the different concentration of the individual species that may be present in a lake water sample. This information could provide one more piece to the puzzle about if your lake is going to get an algal bloom or a harmful algal bloom. An example could be to see if a specific algal species change between the samples taken one month and the next month. Although this represents an exciting opportunity, we do not have any specific examples where we've done this. At this point, it is just an additional piece of information that a lake manager may track to see how it actually correlates with a lake's HAB status. In conclusion, I hope I could convince you that the lake water microbiome presents a number of unique opportunities for lake management. All of the current single microbe indicator tests provide questionable information. In contrast, using the entire collection of lake microbes provides a highly confident information on sources of contamination. And finally, identification of presence and relative concentrations of 150 blue-green algae species could potentially be used to decide when to treat to prevent harmful algal blooms. With that, I would like to give thanks for the opportunity to speak to you. This is an exciting time in developing new solutions through smarter lake management to achieve the highest lake water quality and ensure the health and safety of the users of the lake. We are a new company, and we have a unique approach to provide you with actionable information for your use. I know many of you have questions. The best place to start would be to go to our website listed here.